Go.
In this video, we're going to talk about digestive processes that occur in the stomach. Now, before we get into the processes themselves, let's talk a little bit about the anatomy, the wall anatomy of the stomach. The gross anatomy I have reviewed for you in one of the lab videos on this site. So I'm going to skip the gross anatomy and just talk about the histology. So this organ is going to be in the abdominal cavity, in the left upper quadrant primarily, and it's going to have a couple of specializations that we need to be aware of. First of all, in its mucosa, its epithelium is going to be mostly mucus cells, simple columnar epithelial cells that are mucus cells. And that mucus production is to protect the epithelial surface from the acidic environment in the lumen. Another modification of this organ is that it's going to have more elastic fibers in its submucosa than most other alimentary organs. This is because this organ needs to stretch pretty significantly as it fills up with food. So this is going to give it the ability to stretch and then recoil. The mucosa and submucosa are also going to have folds, which we refer to as the rugae, and you can see those in the model here. And these folds give the organ additional surface area to allow for that stretching as well. Another feature that's different in this organ compared to other alimentary organs is that instead of 2 layers of smooth muscle, we actually have 3 layers of smooth muscle in the wall of this organ. So we have a layer of circularly arranged smooth muscle fibers, a layer of longitudinally arranged smooth muscle fibers, and then we have a 3rd layer of obliquely or angled smooth muscle fibers. And so this gives the organ more ability to move in different directions, and this is what allows for the churning action of the stomach. Okay, now that churning action is important because it is kind of our last backup to the chewing that happens in our oral cavity, as far as giving us an opportunity to mechanically break down the food before we get to the main area for chemical digestion. This is effective as far as mechanical digestion for certain types of foods, but not for all types of foods. Okay, so if you have plant-based material like corn kernel or pea, for example, and that gets swallowed whole, the kind of mechanical breakdown that you have because of the churning of the stomach is not going to be able to compensate for that and break that down further. And you know, the whole purpose, as we've said in a previous video, of mechanical breakdown is it's trying to expose more surface area of the food to enzymes. And if you don't expose that surface area, all of that nutrient content of the food is lost. Okay, and this is a problem in people who don't have good dentition. So that's why your teeth become so important for mechanical breakdown. Okay, the serosa of this organ is going to be typical to the type of serosa we'd find in any alimentary organ. Okay, now we're going to switch down to kind of a microscopic view at this point. So I'm going to remove the front part of this model again. And we were thinking that we were going to be like with the magic school bus and go into the stomach and have the opportunity to shrink down and walk on the surface. We'd see this kind of undulated surface of all of the rugae, and as we looked closer, we'd actually see that in the mucosa there are openings, and those openings are the gastric pits. And I've used the whiteboard here to depict one of the gastric pits. So this is where, you know, if we were in our little magic school bus trip, this is where we would be kind of walking on the surface, and we would see the gastric pit over here. There should be material coming out of that gastric pit that's produced by the cells in this area, which are the cells of the gastric gland, okay? And the gastric gland is going to have different substances that are going to be in that secretion, and the secretion of the gland is referred to as the gastric juice. Okay, so we'll indicate the gastric juice coming out. Okay, so if we were thinking of food material coming in to the stomach that would be delivered through the esophagus, that bolus that we produced in the oral cavity, once it mixed with the gastric juice, would become what we call chyme. Okay, and the chyme is going to be more liquid than a bolus because of the amount of gastric juice that is going to be added. Okay, so what is in the gastric juice that is important to digestive activity? Okay, well, it's the secretions of multiple cells, and we're going to look at two main cells here. The first one that we're going to talk about is the cells here that look like they have more surface area, and these are parietal cells. Okay, the parietal cells produce all of the non-enzymatic components of the gastric juice, non-enzymatic and non-hormonal. Okay, so one of the secretions of parietal cell is hydrochloric acid. Okay, so it's the parietal cells and their secretion that give the gastric juice its acidic pH, typically maintained at around a pH of 2. Okay, and the reason that the hydrochloric acid is important is because it denatures proteins. Okay, so proteins have complex three-dimensional shapes, okay, and they're in kind of a globular form most of the time when we consume them. But anything that's in a globule is going to be more difficult to break down enzymatically because the enzymes are going to have to chew all the way down through the layers to get to all of the nutrient content. So the more that we can spread the proteins out, the easier that chemical digestion becomes. Okay, so denaturing proteins spreads the proteins out, and this is considered a type of mechanical breakdown. Okay, another component that's produced of the gastric juice that's produced by the parietal cells is intrinsic factor. This is a substance that allows us to absorb vitamin B12 in the small intestine. Okay, and so this is very important to allow us to get that nutrient. And if you have deficiencies in intrinsic factor, that can lead to deficiencies in vitamin B12. The other cell that we're interested in here is the chief cell. The chief cells produce the enzymes that are found in the gastric juice, okay? Or in one case, the pre-enzyme, okay? So we do have a type of lipase that's produced and released as part of the gastric juice, but just like with the lingual lipase, this lipid digesting enzyme is not going to be very efficient because there's nothing in the gastric juice that causes lipids to break up. So you have that same issue of the enzymes just kind of chewing from the outside and not being able to get to the core of the lipid droplets. Okay. The other product of the chief cells is pepsinogen, which is a precursor version of the enzyme pepsin. Okay. Now, pepsin is a protein digesting enzyme, and as a safety feature for cells that produce protein digesting enzymes, all of these enzymes are created in a precursor form that is inactive. And this just makes sense if you're thinking, here's this cell here, if it creates an enzyme that can digest proteins, then all of its proteins could potentially be destroyed before that enzyme has a chance to get out of the cell. Okay, so instead, the cell produces the precursor form, and that's the form that's released. Okay, so we'll put the pepsinogen here in the lumen of our gastric gland. That's going to go out into the lumen of the stomach, and it's going to get converted to pepsin in the lumen of the stomach. Okay, and so in the stomach, we will have the initiation of protein digestion. Okay, now there's no carbohydrate digestion that happens here because one of the proteins that gets denatured when it comes into the stomach is salivary amylase. Okay, so because that protein is denatured, that enzyme is denatured, and we don't have an enzyme that is produced here that digests carbohydrates, we don't have any carbohydrate digestion here in the stomach. Okay, so to summarize, in the stomach, we are denaturing proteins. We denature salivary amylase, so carbohydrate digestion ends. We produce gastric lipase, we get a little bit of lipid digestion, and we produce pepsinogen, which is converted into the active form pepsin. That begins the digestion of proteins in the lumen of the stomach.